Hello viewers, welcome to our channel. In this video, we will discuss some important questions on translation. Translation is the process in living cells in which proteins are produced using RNA molecules as templates. The generated protein is a sequence of amino acids. This sequence is determined by the sequence of nucleotides in the RNA. For CSR NET, ICMR NET, GATE, NEET exam, the questions regarding this topic is very important. Some brief notes regarding these questions and their related options are also provided here, which will be beneficial for you. So stay tuned in this video. The first question, tRNA acts as option 1 adaptor RNA, option 2 messenger RNA, option 3 structural RNA, and option 4 none of the above. And the correct answer is option 1, that is adaptor RNA. Transferred RNA, that is the tRNA, is an adaptor molecule that is composed of RNA, typically 76 to 90 nucleotides in length. It serves as a physical link between the mRNA and the amino acid sequence of the protein. The tRNA genes from bacteria are typically shorter than the tRNAs from that of archaea and eukaryotes. The next question. Which of the following act as ribozyme in peptide-bond formation in bacteria? Option A 1. 23S rRNA, option 2. 16S rRNA, option 3. 5S rRNA, and option 4 is 28S rRNA. This rRNA means the ribosomal RNA. Ribosomal. Okay. Ribosomal RNA. Now the correct answer is option 1, that is 23S rRNA. 23S rRNA is a component of large ribosomal subunit of bacteria and archaea and make up the peptidal transferase center. So this 23S rRNA is a component of large ribosomal subunit of bacteria and archaea. This 23S rRNA acts as ribozyme in peptide bond formation. Numerous mutations in the domains of 23S rRNA with peptidal transferase activity have resulted in antibiotic resistance. Next question. Transcription and translation can be coupled in option 1 bacteria, option 2 archaea, option 3 plants, and option 4 is animals. And the correct answer is option 1, that is the bacteria. Coupled transcription and translation is the hallmark of prokaryotic gene expression. So this coupled transcription and translation is the hallmark of prokaryotic gene expression. Coupled transcription and translation occurs when the ribosomal associated with an initiate translation of mRNA whose transcription has yet not concluded, therefore forming RNAP, that is ribonucleoprotein. Next question. tRNA looks like option 1 clover leaf, option 2 L-shaped, option 3 inverted L-shaped, and the option 4 is both 1 and 3. And the correct answer is option 4, that is both 1 and 3. That is tRNA looks like clover leaf structure and inverted L-shaped structure. Now all tRNA conform a secondary structure described as a clover leaf. So the secondary structure of tRNA is a clover leaf structure. And this clover leaf structure has 3 hairpin loops. And it again folded in 3-dimensional space into an upside down L-shaped molecule, that is the tertiary structure. So the tertiary structure of the tRNA is inverted L-shaped, that is the upside down L-shaped molecule, whereas the secondary structure of tRNA is a clover leaf structure. In the tertiary structure, that is the upside down L-shaped structure, the amino acids and the anticodon are opposite ends of the molecules. The molecule of tRNA is folded due to its folding of some complementary nitrogenous base that come across each other and form hydrogen bonds amongst them. Next question. UTRs, that is the untranslated region, is required for efficient translation, which is present in option 1, 3' end of mRNA, option 2, 5' end of mRNA, option 3, 3' end of tRNA, and option 4, both 1 and 2. And the correct answer is option 4, that is both 1 and 2, that is UTR, that is the untranslated region, is present in both 3' end and 5' end of mRNA. The 5' and 3' UTR, that is the untranslated regions of mRNA, are domains that control the critical post-transcriptional gene regulation process. The UTRs are present at both 5' end before the start codon and at 3' end after the stop codon. They are required for efficient translation process. Next question. Which of the following combinations are used in translation? Option 1, mRNA, rRNA, tRNA. Option 2, rRNA, hnRNA, tRNA. Option 3, mRNA, hnRNA, tRNA. Option 4, rRNA, mRNA, tRNA. And the correct answer is option 1, that is mRNA, rRNA, tRNA. Messenger ribonucleic acid, that is the mRNA, is a single-stranded molecule of RNA that corresponds to the genetic sequence of a gene and is read by a ribosome in the process of synthesizing a protein. The transferred RNA is an adaptor molecule composed of RNA, typically 76 to 90 nucleotides in length. It serves as a physical link between the mRNA and the amino acid sequence of proteins. tRNA's genes from bacteria are typically shorter than the tRNAs from archaea and eukaryotes. Where is the rRNA, that is the ribosomal RNA, is a molecule in cells that forms a part of protein synthesizing organelle known as ribosome, that is exported to the cytoplasm and help translate the information in the messenger RNA into protein. Next question. This best describes a polysome. Option 1, active site for synthesis of lipid. Option 2, active sites for synthesis of protein. Option 3, active site for synthesis of DNA. Now option 4, all of the above. And the correct answer is option 2, that is active site for synthesis of proteins. Polysomes are formed during the elongation phase when ribosomes and elongation factors synthesize the encoded polypeptide. Multiple ribosomes move along the coding regions of the mRNA, creating a polysome. The ability of multiple ribosomes to function as an mRNA molecule explains the limited abundance of mRNA in the cells. Next question. The order and the sequence of amino acids are defined by option 1, mRNA, option 2, rRNA, option 3, tRNA, and option 4, all of the above. And the correct answer is option 1, that is mRNA, that is messenger RNA. According to the sequence present on the mRNA, the amino acids are produced. That is why the order and sequence of amino acids are defined by the mRNA or messenger RNA. Next question. Which of the following identifies a particular amino acids in its cognate tRNA molecule? Option 1, topoisomerase, option 2, rRNA, option 3, ribosome, and option 4, tRNA synthetase. And the correct answer is option 4, that is tRNA synthetase. tRNA means transferred RNA. An amino acid tRNA synthetase is called tRNA ligase. It is an enzyme that attaches the appropriate amino acids to its corresponding tRNA. It does so by catalyzing the transesterification of a specific cognate amino acids or its precursor to one of its compatible cognate tRNA to form an amino acid tRNA. Next question. What is true about tRNA? Option 1, It looks like clover leaf in three-dimensional structure. Option 2. It binds with an amino acids at its 3' end. Option 3. It has a codon at one end which recognizes the anticodon on messenger mRNA. Option 4. It looks like L-shaped in three-dimensional structure. And the correct answer is option 4. That is, it looks like L-shaped in three-dimensional structure. All tRNAs form a secondary structure that is described as a clover leaf, and this clover leaf structure has 3 hairpin loops. It is folded in three-dimensional space into an upside-down L-shaped molecule, that is the tertiary structure, in which amino acids and anticodons are opposite ends of the molecule. Next question. Untranslated region, that is the UTRs, are present in option 1. Before and after the stop codon. Option 2. After the start codon and before the stop codon. Option 3. Before the and after the start codon. And option 4. Before the start codon and after the stop codon. And the correct answer is option 4. That is before the start codon and after the stop codon. So the UTRs are present in both 5' end, that is before the start codon, and at the 3' end, that is after the stop codon. They are required for efficient translation process. So how can you remember that the untranslated region are present that before the start codon and after the stop codon? So the translation process before the start, the translation process does not continue, and after it is the translation is end, it is stop, the translation process is ended. So this the UTR region is present, that is the untranslated region is present before the start codon. So and after the stop codon, and because after the translation is end, then translation is completed. Okay, thus you can remember this. Next question. In protein synthesis, translocation is initiated with the movement of option 1. tRNA from P site to the A site. Option 2. Peptidal tRNA from the A site to the P site. Option 3. tRNA from A site to P site. And option 4. tRNA from P site to E site. And the correct answer is option 2, that is peptidal tRNA from the A site to the P site. During protein synthesis, mRNA and tRNA are moved through the ribosome by the dynamic process of translocation. Sequential movement of tRNAs from the A site to the P site to the E site is coupled with the movement of their associated codons on the mRNA. Next question. Which of the following antibiotic inhibits the translation in bacteria? Option 1. polymyxin. Option 2. rifampicin. Option 3. streptomycin. And option 4 is cyclohexamide. And the correct answer is option 3, that is streptomycin. Whereas the polymyxin is act by disrupting the membrane integrity, leading to the permeabilization of the bacterial cell. Next is rifampicin. Rifampicin is thought to inhibit bacterial DNA-dependent RNA polymerase. And the third one is the cyclohexamide, that is a protein synthesis inhibitor in case of eukaryotes. Whereas the streptomycin, tetracycline, etc. are protein synthesis inhibitor in prokaryotes. In bacteria, that is the prokaryotes. Next question. This elongation factor is known as translocase. Option 1 EFG. Option 2 EF2. Option 3 both 1 and 2. And option 4 EFTU and EFTS. And the correct answer is option 1 or option 2. That is EFG or option 2, that is EF2. It is MCQ type question. So both answer will be correct. that is option 1, that is EFG, and option 2, that is EF2. Both elongation factor act as translocase. Archaeal and eukaryotic elongation factor 2 and bacterial elongation factor G catalyze the translocation of tRNA and mRNA from amino acid site to the peptidyl site of the ribosome. So these two helps in act as translocase, that helps in the translocation of tRNA and mRNA from amino acid site to the P site. EFTase function as guanine nucleotide exchange factor. This EFTase function as guanine nucleotide exchange factor. It catalyzes the reaction of EF-TU GTP to EF-TU GTP. EF-TU then delivers the amino acid to the ribosome. Therefore, EFT's main role is to recycling the EF-TU back to its active sites in order to complete the elongation cycle. Thank you. If you like this video, please like, share and subscribe to this channel. Thank you.